Okay, guys, welcome back. We have talked about how it is that we can test antibiotics in a laboratory and their effectiveness. We've talked about where a lot of them come from. Other microbes is where we get the idea, right? And many of these ideas come from soil bacteria. But now we're going to look at the mechanisms, the specific mechanisms by which the different antibiotics that we use kill bacteria. All right. Now, this is going to require us to remember that there are differences between bacteria and eukaryotic cells, and we're going to have to key in on those differences. All right. That's the whole thing about really understanding how it is and why it is we might use the drugs that we're going to talk about. Okay. First thing, cell wall synthesis inhibition. If I want something that is selective in its toxicity, right, ideal characteristic of an antibiotic, then cell wall synthesis is a perfect choice, right? Because we don't have cell walls in human cells and in eukaryotic cells that are animal cells, right? So this is a great thing to go after, right? We can go after protein synthesis. Now, this sounds like a problem because of course, we have protein synthesis going on in our cells, in eukaryotic cells, but we also do have differences in that process that we have learned. One of the differences right off the bat, we could say that the ribosomes are different, right? So if we can pick a drug that keys in on a certain size ribosome only found in these prokaryotes, that would be useful. We have nucleic acid synthesis inhibition. Production of nucleic acids, right? So DNA or RNA production. We can look at membranes and disrupt those membranes. Even though membranes are common to all cells, all cells have a plasma membrane at the very least, there are differences in the protein makeup and fatty acid makeup within those membranes. And we can pick drugs specifically for their ability to disrupt the membranes of microbes we want to get rid of. And finally, metabolism. We know for sure there are differences in metabolism between an aerobic mammalian cell like in a human and those in bacteria. And even among bacteria, we know that there are metabolic differences in how they function. All right, so these are the 5 major categories. You're going to want to know the 5 major mechanisms of action that antibiotics have. Right? These are where we find all our antibiotics. And you should be able to classify any antibiotic you look at into one of these 5 categories. All right? Now in your text, and I think this is in, I know it's in chapter 14, I think it's 14.3. 14.3, okay? They have this image, and it's a nice one because it kind of shows you those 5 things that we just got done talking about, right? So we can key in on ribosomes, something to do with protein synthesis. We can key in on membranes, the cell wall, DNA or RNA synthesis, or we can go after the metabolic pathways that are unique to some of these bacteria. Now here you see some examples, right, under each one of these of maybe some antibiotics you recognize, right? Definitely penicillin, you know that one. But a bunch of these you might have also heard of. Cipro, let's see, chloramphenicol, maybe you've heard of that. Tetracycline, right? A lot of cycline drugs. Sulfa drugs, we see sulfo in front of these things, right? So this is a good little figure you can find in that chapter. And please do go and read about it. If something I talk about doesn't quite hit home, it's always good to check out another source, right? And maybe take a look at it there. Maybe it'll make more sense. All right, so let's first hammer home the cell wall antibiotic category. Okay, what I'm going to need you to know in respect to the test coming up, the this would be the third exam, okay, would be, when it comes to antibiotic categories, what they are, right? So cell wall is the first one. But then you'd want to be able to tell me what are the subcategories for antibiotics, okay, that are in this category. And there are 5 groups, okay? So when it comes to inhibition of the cell wall synthesis, the production of the cell wall, we have beta-lactam group, and this is basically the penicillin family, okay? The beta-lactams are the penicillin family, and so if you see "cillin" at the end of these words, it doesn't have to be penicillin, it's probably a beta-lactam. We have carbapenems, and we'll talk about each one of these here on the subsequent slides. We have cephalosporins, glycopeptides, and antimycobacterials. Okay, so we have 5 subgroups within cell wall antibiotics. Now this category of cell wall has the most subgroups, so it gets easier, okay? But these are the subgroups. So let's look at the beta-lactams first. And what I want to bring your attention to here is this penicillin molecule down here. Now I'm not going to have you reproduce these or identify these, but I do want to bring attention to this square right here, the square in red here. This particular subgroup of atoms within this molecule, this is a beta-lactam ring, okay? It's a lactam ring. So what makes the penicillin family, the beta-lactams, unique and effective is the fact that that square is there in all of the cillins, okay? In fact, we're going to find that this basic structure is found in more than just the beta-lactams, all right? But we're definitely going to find that this plays a big role. So here we have a couple cells that are being destroyed because they can't maintain their cell walls. And here we have a little video. Hopefully it works here. Let's see. This is a little video of cells, which definitely look like bacillus cells. And they are growing, but there's some kind of penicillin family antibiotic in here. Pop, right? You can see these cells suddenly explode, okay? And what this is, it's pretty cool, right? This is the effect of beta-lactams or some kind of cell wall drug on the bacillus cell that you saw there, okay? Now, there are natural penicillins. Remember we said there could be natural, semi-synthetic, synthetic, right? So there are natural penicillins, and the original penicillin Fleming found, right, that was from actually a fungus that killed bacteria. A lot of those penicillins were not ideal because they cause side effects, and they're not, the immune system, maybe they had some negative, some drawbacks, okay? But these are some natural penicillins, and sometimes you find that's the case with natural penicillins. And you can see, here's that square that I mentioned to you guys, right? We're going to find that square every time. But with the penicillin family, the beta-lactam family, we're going to have more than just that square, right? We're going to have a common nucleus, they call it. It's not an Adams nucleus. It's a nucleus in the sense that it's a core portion of the penicillin family molecules that is more than just this square, right? But some of these, can only be effective if it's injected. Penicillin down here, penicillin V, is only effective when it's taken orally, right? So there's some drawbacks, some limitations that we have to each of these. Now, by the way, when I go over some of the kind of neat, unique features of these various penicillin family members, I don't need you to memorize every one. It'd be nice if you knew psyllin at the end kind of tipped you off that this is probably a member of the beta-lactam group. But in the exam, I'm far more likely to ask you for an example within the beta-lactam family. Okay? I'm not going to just throw names of antibiotics at you and expect you to know where everyone fits. Okay? So here we have some semi-synthetic penicillins. Okay, so these were taken to the lab and then altered. And you still see the psyllin though, right? So we know that that's a beta-lactam. And we see that, this changes their effectiveness. This is a narrow spectrum drug. This is an extended spectrum drug. Again, I'm not expecting you to memorize that oxacillin is narrow spectrum and what particular bacteria it kills. What I'm expecting you to do is to know what semi-synthetic means. If I were to tell you that this oxacillin is narrow spectrum, I want you to be able to tell me what that means about the drug. Okay. Now there are also 4 other groups, right? Beta-lactams, this penicillin family, is not the only one. We have carbapenems. For example, imipenem would be an example, and you will want to know an example for each subgroup, okay? So we've got 5 categories. In this particular category, we have 5 subgroups. You'd want to know the subgroups. Carbapenem is one, and imipenem would be an example. Now, it just so happens that imipenem is actually a broad spectrum. Let's see, I've got some notes here. And again, some of these details about each drug that is just for your information and just kind of to make it more interesting. A lot of carbapenems are bactericidal. So this is another twist on this particular subgroup. We have another subgroup that's a cell wall antibiotic called cephalosporins. Now, an example I've got here, you can remember that one, but there's tons of other drugs that are in this category. And most of them start with what sounds like ceph, okay? So C-E-P-H, some of them are spelled with C-E-F, some with K-E-F even, right? So if you see those starting the word, it gives you a little tip-off as to what subgroup and category it belongs to. A lot of these cephalosporins have fewer allergy problems related with them compared to some of those beta-lactams we talked about. So you can see where we might pick and choose within the subgroups based on maybe an allergy somebody has or something like that. Now I just thought I'd show you, I've gone through 3 of the subgroups within the within the cell wall category. I thought I'd show you a picture of what these look like. Again, I'm not going to throw pictures of these at you, but I just want you to know, just to drive home, the beta-lactam ring. So here we have penicillin. We expect to see a beta-lactam ring. There it is. Okay, but we expect to see a lot more than that in a beta-lactam or penicillin family molecule. It's a common nucleus, this whole deal here, right? But when we look at imipenem, which is in a different subgroup, guess what? We still see a beta-lactam ring. But there's more that's happening here, right? The common nucleus that we see with penicillin, well, that's all changed, right? So again, it keys in on the fact that beta-lactam rings are really important to inhibiting something about the bacterium that protects itself from antibiotics. Here's another cephalosporin, right? Now that's a different group, right? We have beta-lactam group, penicillin family. We have carbapenem family. We have cephalosporins. And it also has the beta-lactam ring. This beta-lactam ring is huge, okay? This shape enables the molecules to inhibit enzymes that protect the cell. Okay? So it's all about this it's either a competitive or a non-competitive inhibition. They don't know for sure, but it definitely is helping to kill the cells that are causing infection. Okay, a fourth subgroup. So we've got penicillin family, carbapenems, the cephalosporins, right? Now we have the glycopeptide group. Now this contains an example known as bacitracin, and I said I'd visit this again later. One of the organisms we tend to find antibiotics are produced by, and we steal these ideas, is Bacillus species. Bacitracin happens to be one of those stolen ideas. The original chemicals that gave us the idea for bacitracin come from Bacillus species. Now, bacitracin happens to be narrow spectrum against gram-positive organisms. Another glycopeptide would be vancomycin. You might have heard of vancomycin before. Also narrow spectrum against gram-positives. And a lot of these kill cells outright. Again, I'm not so concerned you memorize that they're narrow spectrum or bactericidal. I would say if I gave you that information, you'd want to know what that means, okay? But just some interesting information I want to add. Bacitracin happens to be somewhat toxic in the system, but it's very effective against bacteria. It goes after those cell walls pretty well. The fact that it's toxic means that we don't take these things orally, okay? This is usually used as a topical application on the surface of maybe the skin. It's used for eye ointments to kill bacterial infections on the surfaces, okay? Vancomycin, this was originally, when developed, a really toxic drug. It had some pretty nasty side effects, but over the years, purification of the drug has improved and fewer side effects are associated with it. So all of these drugs have gone through an evolution when we find semi-synthetic and synthetic forms of the drugs, and just purification alone will help to avoid some of the side effects we want to avoid. Okay, finally, our fifth subgroup within the cell wall category are antimycobacterials. Now, I'd like you to just spend just a second here. And anti, we know what that means. It's against, right? But mycobacterial. Mycobacterial. What if we said mycobacterium and stuck tuberculosis after that? Okay? We are talking about a group that has a specific kind of cell wall. We learned about acid-fast stains, the fact that they would show us tuberculosis in a sputum sample. Well, here, I mean, you remember, right? They have a specific kind of cell wall, kind of a lipid characteristic in their cell wall. So here we have antimycobacterials. These drugs are effective against those kinds of organisms, like tuberculosis. Okay? Here we have an example, ethambutol, and it has a narrow spectrum against what? Exactly what you'd expect. This unique group that we learned about that causes tuberculosis and leprosy and stuff like that, right? It inhibits the incorporation of that kind of fatty acid, lipid molecule, mycolic acid, into the cell wall, all right? And it's usually bactericidal against those particular bacteria, okay? All right, so we'll call it quits at this point. That's our first category of the 5 antibiotic categories we have. You'll want to know the name of the category, the subgroups, an example of a drug in each. Okay? I'll catch up with you next time. We'll talk about our next category.